Our bodies are capable of incredible things. From simple movements to complex athletic feats, the human body is a marvel of engineering. Muscles are the driving force behind every action we take, big or small. Skeletal muscles are made up of bundles of long cylindrical cells called muscle fibers. These fibers enable us to perform a wide range of movements. Think of these fibers as long, thin threads that are bundled together to form a muscle. Each bundle allows for efficient and powerful contractions. Each muscle fiber contains smaller threads called myofibrils, arranged in a precise pattern, contributing to the muscle's ability to contract and generate force. Myofibrils contain the essential proteins actin and myosin, which interact to produce the force needed for movement. The sarcoplasmic reticulum stores and releases calcium ions, triggering the interaction between actin and myosin, leading to muscle contraction and movement. Myofibrils generate the force of muscle contraction. They are made up of sarcomeres, the basic contractile units. The interaction between these filaments drives muscle contraction. Sarcomeres are the fundamental units of muscle contraction. They shorten muscle fibers, leading to contraction and force generation. Sarcomeres are defined as the region between two Z lines. Section 5. Thick filaments, the myosin powerhouses. Myosin is fundamental to muscle function and movement. Thick filaments are primarily composed of myosin, the driving force behind muscle contraction. Myosin molecules resemble two golf clubs twisted together, essential for muscle contraction. The head region binds to actin and ATP, crucial for muscle contraction. Myosin heads attach to actin, forming cross bridges, pulling thin filaments, generating force and enabling movement. Section 6. Thin filaments. Actin and its regulatory proteins. Thin filaments, composed primarily of the protein actin, are essential for muscle contraction. Tropomyosin and troponin regulate muscle contraction by controlling the actin filament. Section 7. T-tubules, the electrical messengers. T-tubules are extensions of the muscle fiber's plasma membrane that penetrate deep into the cell. They transmit the electrical signal for muscle contraction, ensuring all parts of the fiber contract simultaneously. Section 8. Sarcoplasmic reticulum, the calcium storehouse. The SR stores and releases calcium ions, essential for muscle contraction. It surrounds each myofibril, forming a network of sacs and tubules. Section 9. The dance of muscle contraction. Muscle contraction is a fascinating process involving proteins and ions. A signal from the nervous system triggers calcium ion release. Calcium binds to troponin, exposing myosin-binding sites. Myosin heads attach to actin, forming cross bridges. Myosin heads pivot, pulling thin filaments, causing contraction. Section 10. Conclusion: muscles in motion. Skeletal muscles are marvels of biological engineering, allowing us to move with precision and power. Understanding skeletal muscles deepens our appreciation for biology and has practical implications for medicine, sports science, and robotics.